is primarily an X-ray crystallography lab. Most of the structures we do are by crystallography. So this talk is going to start and end in crystallography, but I will mention kind of some of the work that we're doing to introduce cryo-EM into the lab, which is something we obviously have to do to tackle the larger proteins that we can't do with crystallography. So we go forward. Strong press. Right. So I'm going to start with discussing some of what we've done in structural biology and MAB isolation to impact immunogen design for HIV-1 and SARS-CoV-2, reaching back to my postdoc, if that's okay. And then carry on to the more exciting stuff that we're doing now, developing snakebite therapeutics for snakebite inflammation, next generation therapeutics. And this is a really tough button to push. Okay. Focusing first on HIV-1. And so kind of what we do there is something called reverse vaccinology 2.0. So this is some term that was invented by Dennis Burton. And so the idea here is you look at really broad and potent monoclonal antibodies that bind to a particular antigen. You use structural biology to figure out how they bind, and then you redesign the immunogen to be able to elicit those antibodies through vaccination. And so, like I said, this is work that I did at Scripps as part of my postdoc. And so just first introducing this molecule, which is the envelope trimer of HIV. The yellow bit at the top is the bit that binds to the host receptor, and the red bit's the bit that promotes membrane fusion. And so this is the protein that's on the outside of the HIV virion. So if you think about vaccines and sterilizing immunity, the traditional way that we think about that is neutralizing antibodies. Neutralizing antibodies are the things from the immune system that come and stop the virus from infecting the cell. And so this HIV envelope is the sole target on this particular virus for neutralizing antibodies. And if we kind of just paint over somewhat artistically to show two different types of epitopes that are on this envelope, we first have in light blue what are known as hypervariable regions. So these are kind of the most accessible pointy outer bits of the envelope that are bound by strain-specific neutralizing antibodies. And those antibodies are strain-specific because these sites are very sequence diverse. There's basically very little overlap from one individual to another. And so these types of antibodies are going to neutralize just one or two viruses from a single individual and nothing much more. And then in the rainbow colors, these are kind of more assessed conserved epitopes for what are known as broadly neutralizing antibodies. And for those people that are interested in HIV vaccine design and think that that's something that may one day work, these are the epitopes that you would want to elicit in an HIV vaccine. And so for this talk, I'm just going to focus on the top most region of HIV, which is known as the V2 apex, where you can see there's a collection of both strain-specific and broadly neutralizing antibody epitopes in light blue and red. So this is a single crystal structure of one V1, V2 monomer. So in cyan and yellow, we have those outermost pointy bits, the hypervariable loops. And if you look over at the sequence alignment on the right-hand side of the slide, you can see, again, with the cyan and the gold colors, the sequence alignment that corresponds to a hypervariable loop. And you can see there's literally no sequence overlap between the 5 representative strains on the slide. And then going back to the crystal structure, the green bit is the conserved portion that broadly neutralizing antibodies would recognize. And again, looking at the sequence alignment underneath strands B and C in green, you can see there's a lot more conservation, and that's why these are susceptible to broad neutralization. And a lot is known structurally about how broad neutralizing antibodies actually bind to these sites and affect this broad neutralization, but very little was known about strain-specific neutralizing antibodies and the epitopes that they target. And so what we did was solve a bunch of crystal structures of different strain-specific V2 neutralizing antibodies bound to V1, V2. And you can see that in all these cases, the antibodies are coming in with the same kind of angle of approach, the same kind of lateral. I don't know what's the pointer because there's two screens, but they're all on the left side of the V1, V2, right? And so to take that and then compare it to epitopes for broadly neutralizing antibodies, we can kind of divide this single V1, V2 monomer into a strain-specific neutralizing antibody face and a broad neutralizing antibody face. And so that's divided by the in pink, the two glycans, the 156 and 160, a little glycan fence. And that can then kind of take us to immunogen design, where we're looking now at the HIV envelope trimer from the top down. So this is from the apex view. And you can see that there's 3 clefts between the 3 different V1, V2 monomers. And we can break up each of those clefts into a strain-specific neutralizing antibody face and a broadly neutralizing antibody face. And if we then plot center of masses for a bunch of known broadly neutralizing antibodies and the strain-specific antibodies that we solve, you can see that they all kind of divide themselves under that or over that dashed line, and that dashed line represents the difference between the BNAb face and the SSNAb face. And all the broadly neutralizing antibodies are binding on the SSNAb face, on the BNAb face, and all the strain specifics above the dashed line. And similarly, if we flip the structures, and so looking at center of mass relative to a line going from the trimer apex down to the membrane, we have this arbitrary 45-degree angle where you can see all the BNAbs are binding above that 45-degree angle, where all the strain-specific antibodies are kind of below that angle. And so this gives us some idea on how to modify HIV immunogens through glycan masking configurations to kind of guide the immune system towards these spaces where the BNAb antibody precursors need to come in and hide away the strain-specific neutralizing antibody faces. And that's important because strain-specific antibodies in purple and broadly neutralizing antibodies in red to the trimer apex cannot coexist at the same time. The one binds, it prevents the other one from binding. And so if in a vaccine immunogen, you would then lose out on the opportunity to start getting BNAb precursors going. And so that was all a bunch of work I did in the Wilson lab. And then I was done with HIV and thought I needed to work on something that for me was somewhere where I felt we could really make a real difference in the world. And that was snakebite antivenom therapeutics. And so November 2019, I came back to South Africa to work on this topic. And then unfortunately, the world said no. And we had SARS-CoV-2. And so like everybody else, I had to pivot slightly towards some SARS-CoV-2 research for a little bit. And kind of my introduction into the SARS-CoV-2 field, I just put this slide in to illustrate a point. So the introduction was with the advent of variants of concern. And so this work done here didn't actually involve generating any new structures, but it just shows how understanding protein structure and being able to interpret protein structures really already can have a massive impact on public health. So this was shortly after the Beta variant, which was the first variant of concern, started to emerge in the Eastern Cape. And we looked at a plethora of structures that had already been done over the year of 2020, looking at all the MABs that bound to the original Wuhan strain from SARS-CoV-2. And just looking at those structures, you could see that the few mutations that Beta had picked up were all resistance mutations to all of these known neutralizing antibodies. And so we were able to predict very rapidly that the Beta variant was going to be resistant to neutralization by plasma antibodies, both from infection and vaccination. And then collaborating with Penny Moore's group in the ARI unit, we were able to, within a month, prove that that was in fact the case. So just, highlighting the importance of structural biology in informing public health. So we've continued to collaborate with the Moore group. And so this is an effort that they have towards isolating broadly neutralizing anti-sarbecoviruses. These are monoclonal antibodies that can neutralize a bunch of different sarbecoviruses, which are bat-related viruses to SARS-CoV-1 and SARS-CoV-2. And so just the top table, which is kind of a heat map of some of the antibodies that they've isolated, where the strong colors on the left-hand side show a lot of broad neutralization. And so we picked the top two antibodies from this table to kind of do some structural biology on and figure out how exactly are they binding, what makes them broad and potent. And so we started with two crystal structures. So these are crystal structures of the two most broad antibodies, E4 and C4, bound to RBD in yellow. And the first really interesting point was that these are unrelated antibodies. So they come from different V genes. They were differentially elicited during this infection, and yet they're binding to the exact same epitope. They're coming in, the blue and the red antibodies, with the exact same angle of approach. They're binding to the same sites. They're binding to the important 343 glycan. And that was striking. So it shows this really structural convergence on a single vulnerability across all different sarbecovirus strains. And then the other really interesting thing, which is shown by the orange arrows on the right-hand side of the slide, is that only half the antibody paratope was engaging with a single RBD. So you can see the light chain is engaging, part of the CRH3, but we had this gap where CRH1 and CRH2 was doing nothing. And so that kind of told us we weren't seeing the full picture with crystallography, and that's why we had to turn to, I guess, the topic of this meeting, cryo-EM, woohoo, go cryo, to see the full picture. And so this is in collaboration with a colleague up in Lausanne, where we traveled up there and did a cryo-EM structure of E4, the most potent antibody, onto a full SARS-CoV-2 spike. You can see it comes in with the same kind of from the top apical approach as V2B NAbs and HIV would do. And from these data, we were able to show, if you remember the arrow pointing on the left-hand side of the red antibody, that the CDRH1 and CDRH2 are in fact interacting with an adjacent RBD from an adjacent protemer in the trimeric SARS-CoV-2 spike. So it helped us to explain that, which is the importance of seeing these big molecules by cryo-EM. And that's just a top-down view over there. You can see how the antibody is coming and bridging these two different RBDs and really locking the RBDs down in the spike, preventing them from coming up and interacting with the ACE2 receptor. The other cool thing about the structure is that we see that when the antibodies come in and bind these RBDs together, they kind of pull the spike tighter and push out the other floppier domains of the spike kind of out and downward. And so that kind of gives us some insight into immunogen design for pan-sarbecovirus vaccines, where if this was the kind of epitope which is well conserved that we wanted to kind of elicit antibodies to, we might think about using biodirectional design to tighten up these spikes, prevent them from popping out, but also bring them closer together to make this site more immunogenic for precursors for these types of antibodies. That's the virology done. And that brings me with much excitement to the current major push in our lab, which is monoclonal antivenom discovery and next generation antivenom therapeutics to treat snakebite envenomation. And this very cool topic. And many people ask me, why snakebite? And so I tell them that snakebite is the biggest public health problem that they've just never heard of. It's one of the biggest crises around the world. It's considered the most neglected of WHO's neglected tropical diseases. I have some sinus issues. And so roughly one person dies every 5 minutes from a snakebite around the world. It's really a massive crisis. 4 or 5 times as many people lose a limb, suffer from some kind of disability and disfigurement. And despite all of those data, snakebite is something that we treat essentially with a crude, outdated technology, which is horse blood, right? So there are no modern antivenoms for this massive public health crises in the developing world. Current antivenoms are animal plasma. They're really batch-to-batch variable because they're coming from different horses where the blood is pulled together. Only 10 to 20% of the product is actually active antivenom. The rest is just junk foreign horse protein that essentially sends you into anaphylaxis. So you have to give antivenom in a hospital because the antivenom can kill you. It's very expensive. People are selling their houses. People are selling their livestock and their land to be able to afford this antivenom, and then they're stuffed afterwards. And so we think that these monoclonal antibodies are really the answer to that. And so next generation antivenoms are pure, obviously. We can make them in the lab. They're super safe because we can make them human. They're reproducible. We can make them more potent and more stable through structural biology and rational design. And we think they can be significantly cheaper. So something down to like $5 or $10 for a therapeutic dose, which is really affordable compared to $200 a vial. And you need multiple vials for antivenom therapy. So this is our approach, learning from infectious disease research. So essentially, we're isolating monoclonal antibodies from antivenom horses, and then using structural biology to understand how those antibodies bind to the snake toxins and affect their antivenom effects, and then use structural biology to further improve those antibodies and make them into super potent and broad biologics. That being said, venom is a complex target. There are something like 50 different proteins and their isoforms that the snake injects into your blood once it's envenomated you. They're all doing different things. But we can kind of broadly class the major toxins in snake venom into 4 major classes. And so the idea is to develop 1 or 2 monoclonals to each of those toxin classes. So we end up with something like a maximum of 8 antibodies. We combine them to some sort of bi- or tri-specific therapeutics, and then make a cocktail that we can give to somebody that would then hopefully treat multiple snakes, perhaps within a single geography like within Africa or with South Asia, but generally we think we can get much broader than that and have a single cocktail that we can give to somebody who's been envenomated by any number of snake species. And so this is some of our early data where we've just been focusing on a single toxin class. So these are 3-finger neurotoxins, which are the most deadliest of the toxins. Essentially, these are the ones that kill you within 20 minutes or a couple of hours. And so we've been isolating some super broad and potent antibodies to this particular toxin class. And this is just a summary because I guess my talk is wishy-washy all over different topics, and I didn't go into one specific field. So this is just a summary of some of the broadest antibodies that we've isolated so far, antibodies 3A and 3D. So they're somatically related, they bind in the same way, and they have this breadth across two different subclasses of these alpha neurotoxins, which are called short chain and long chain neurotoxins. And so this is the first in the world. There's a couple other labs that have been trying to make monoclonal antibodies, and they only have breadth in the long chains. So these are the first antibodies that are binding across long and short chains, and they're super potently neutralizing both of those toxin classes as well, which you can tell from the neutralization curves underneath this kind of heat map. And also kind of a first in the world, these antibodies are neutralizing whole venoms. So we can take in an in vitro assay venoms from snake and look at just their neurotoxic component, and these antibodies are completely neutralizing that neurotoxic component in both spitting cobras, non-spitting cobras, and mambas. So really diverse different types of snakes. And this is an example of what that breadth looks like on a sequence plot. So essentially, at the top, we have the long chain neurotoxins clustering in one box, and at the bottom, the alignment and the box of the short chain neurotoxins. And if we look down on the heat map, kind of the bottom left or the top right, you can see these antibodies are recognizing toxins that have as little as 29% sequence identity between them. So really sequence diverse toxins and super broad antibodies that are able to recognize these two different subclasses. So being structural biologists, we wanted to know, how do these antibodies do what they do? Why are they so broad and potent? And so we solved crystal structures of antibody 3A bound to a black mamba short chain neurotoxin, and antibody 3D bound to a consensus short chain neurotoxin. And as I said, they're somatically related, so we expect them to bind in the exact same way. But what we see is that they're binding between fingers 2 and fingers 3, which is an important location for the binding of this toxin to the N-acetylcholine receptor, which is the host receptor where these toxins are having their effect. And they have different ways of improving that binding, so I actually didn't include that slide. But the antibody on the left, 3A, you can see has a slightly longer CRH2 loop over here, not present in 3D. And that loop is what adds an extra bit of binding surface to give the antibody extra affinity. And when we looked at 3D, we found that, in fact, it didn't have that, but it had another insertion in CRH1 that allows it to bind toxin dimers. We don't know the physiological relevance of those dimers, but it seems like it might be another way that it's able to increase its affinity through these somatic up mutation loop increases. But going back to talking about how the antibody recognizes the receptor binding sites, so in purple and blue, in cyan, we have the two sites that the N-acetylcholine receptor recognizes on this toxin. So between finger 2 and finger 3 is the purple epitope, which is bound by loop F. And that's kind of the lesser studied interaction between this toxin and the host receptor. And then on the right side, we have the most common studied one, which is this loop C interaction between finger 1 and finger 2. And in fact, those antibodies I mentioned from other labs that have really good broad antibodies against long chain toxins, they're all binding in that site between finger 1 and finger 2 and mimicking that loop C interaction. And so what we have is something completely new, where we're binding almost exactly the same residue. So if you look at the sequence alignment at the bottom, the purple dots and the kind of hot pink dots, there's almost exact overlap between the epitopes for antibodies 3A and 3D and the loop F binding site from the N-acetylcholine receptor, except for maybe one or two exceptions. So that explains the breadth. It explains the ability to bind short chain neurotoxins, which previous antibodies don't do. And it also gives us now a way that we're exploring. I don't have the data yet, but it's looking pretty promising, where we're using what we know about interactions with loop C to add additional contact sites with the antibodies that are just binding in the loop F binding site to make them even broader and more potent. And so obviously we can only do that because we understand the structures and where they bind. And I like to give short talks. So that is me. I have to thank Mashilo, who did a lot of the initial cloning expression work in the lab and then went off to better celeries. Francis Ayres, a PhD student who did some of the 3D structure work, and then Tyler Hutton has been doing a lot of cloning and stuff as well, and ELISA screening of breadth. I have Ian Wilson up there for the HIV work, running an area team for the SARS-CoV-2 work. Alex Antonisevich at EPFL, where we did the cryo. Nick Rohit and Cassie at LSTM. So those are our collaborators that did the in vitro venom neutralization assays on our antibodies in the UK. IO4B scientists, where we collect all the crystallography data. And then the funders. So Morris Animal Foundation, which funded the initial MAB isolation setup pipeline. I was a Flair Fellow at the Royal Society when I initially came back to South Africa. And I had an NIH-R21. And so a lot of the funding went into initial isolation and structural characterization of MABs 3A and 3D. And Wellcome have agreed to continue funding this work for the next 8 years. And that's me. Thank you.
You got a lot into your short talk. Well done. Thank you very much. Excellent. Okay, so any questions for Kurt? Come on, guys. Okay. Is somebody going to take her? Okay, I'll bring you to you. Come on.
Thanks, Kurt. I've seen the data before, but I'm always confused about one thing. So the E4C4, right? They bind to the two RBDs, but when you crystallize it, it was just one. So I'd assume with the two, it's a stronger affinity. So why do you think it crystallized just with one and not the two RBDs?
That's very strong.
And here we are. The map I've got you run backwards and forward.
Yeah, obviously the interaction with the first RBD is much, much stronger. There's a lot of polar contacts there. There's a larger surface area that's bound. I don't think there's enough surface area contacted on the second RBD that would actually facilitate a single structure with that RBD. And when we look at the contacts that we find in the cryo-EM, a lot of it is primarily just complementary van der Waals interactions, nice little kind of lock and key fit in how the two come together. But there's no real key polar interactions or anything like that would drive the affinity between that interaction.
Yeah. We have another question at the back.
Hi, Kurt. Thank you so much for this unique work on antivenom. What is the linkage between horse antibody and that snake venom antibody you mentioned that you're extracting from horse and then you using it for that? I think I missed that part.
What is the connection between?
Yes. How are you linking?
Right. Yeah. No. So that didn't show the whole pipeline. Right. So essentially, antivenom is made in horses, right? And so we use the horse because it's been hyper immunized for years. It's making these fantastic antivenoms. All the other labs are doing phage display from rough libraries or whatever, and so there's not that element of somatic hypermutation that we're able to take advantage of. So we go in and get the B cells from these hyper immunized horses, and then do single memory B cell isolation of those antibodies. And then so once we've got the genes out of each of those antibodies, we can screen them to look for the broad antibodies. And then once we've picked the best, those are the ones that we follow up with all the functional data and structural biology.
Regarding the antibodies against snake bites, what would be a typical scenario for clinical trials once you have the antibodies?
So it's a difficult one. I think the general theory is that because it's an NTD, there's just no money for clinical trials. I don't know if clinical trials are going to happen. If they do, it'll be a very small scale kind of thing, maybe in South Africa. But we're kind of hoping or leaning towards more of a compassionate use scenario where we show in preclinical models in the mouse and in the in vitro assays that these antibodies are really good, and then go into like a dose de-escalation or compassionate use type of scenario with the antibodies. So I will say we're currently trying to patent these first couple of antibodies, and so the idea there will be to maybe include them with current antivenoms already as a way to try and make current antivenoms more effective, to use less, make them cheaper. I don't know if that's going to pan out, but over the next couple of years, we should have some really good antibodies against all the other toxin classes as well. And then we can start to see how we can get the cocktails into the field. But I don't know that there will ever be a clinical trial of any of these products. It's been difficult enough to try to organize one with current antivenoms as it is. So.
Okay, got. Yeah, I think I've got two questions for you. And my first question is, you talked about the interprotomal contacts, when you were showing that slide on the HIV work. I just want to know, uh, how much of conservation do you see that actually you've got to work through to be able to have a sort of structure-based design of, new therapeutics on that? That's one. And then the second question is, is there any relationship from your work on the snake venom, as you know, that you can translate even into scorpion venoms?
Okay. When people ask you questions, it's always easier to remember the second and tackle that one first. Yes, there are other animal models or horses, even at SAVP in South Africa, that have been made for scorpion venoms, and that is something that we kind of are looking to tackle on the side. But snakes are the ones that are doing the real damage in the world, and that's where most of our focus is initially. And then I'm not quite sure I understood your first question.
I'm just asking about, is there any sort of conservation, as in when you look at the, that site of interaction, that contact that you showed during when you talked about the interprotomal contact. So I'm asking, what really do you see as regards the residue conservation? So, and how you can basically advance that for, design of new therapeutics.
Sequence conservation in the viral immunogens, right? So there's obviously some level of conservation wherever there's interprotomer contacts. So the antibodies that were bridging two protomers were SARS-CoV-2 antibodies, and in that particular instance, the two RBDs are not tightly bound to each other because they have to, for function, be able to flip up quite readily. And so there's not a lot of contact there. And in fact, when we talk about therapeutic design, I think the major key contacts are on just the first initial RBD. And that's actually part of why the antibodies are so broad, because they have this kind of van der Waals interactions with that second protemer that allow them to lock down the spike. But at the same time, they can tolerate a much higher degree of sequence diversity at that particular site. And that region is slightly more variable than the contact site on the main RBD protemer. So it's kind of a way to allow for some flexibility that then allows for antibody breadth. And so you wouldn't really want to mess with that too much, because if you tightened in and focused on that site, you might actually then end up making a less better antibody.
Thank you.
Okay.